I cannot.
Okay, so Peter, I think we are ready to start. Just let me know.
We are ready to start. And I don't know what to do how to start. Okay? But now I'm not visible. Okay, but I'm visible. Okay. So it is 12 o'clock. So we start. Thank you very much. Thanks very much, everybody, wherever you are, for joining this meeting of the NETSOB, the International Network on Soil Biodiversity. Good morning, good afternoon, good evening, all of you. As a board, we welcome you very much, and we try to prepare an agenda for you, which I hope will be inspiring and informative. This agenda will include a keynote, where we will start with a keynote, get progress report, and also we have the election of a new chair of NETSOB. That brings me also to those who have missed the new agenda. There's a change in the original agenda. The election of the new chairs in NETSOB will be tomorrow at 12 o'clock. So not at 1, but at 12. If you want to be part of that item of the agenda, please log in at 12 o'clock tomorrow. We have a meeting of 2 times 2 hours, and each meeting also will have a short break, so you can have a relaxed moment halfway the meeting. There are also some logistical remarks I want to make. And the first is that this meeting is recorded. And the second is that we have a lot of participants registered, close to 1,000, I've understood, so that's fantastic. But we have only little room for having questions or remarks. We will do our best to get some of those addressed. But if you have a question or remark, please do it on the chat. If you don't have any time to address it during the meeting, we have stored it in the chat so we can respond to your questions or remarks later. So don't worry about anything that we don't have time to discuss, because it will be all recorded and stored in the chat. Having said that, I hope that we will have a very informative, fruitful, and inspiring meeting. And as a starting, we would like to give the floor to.
No, sorry, Peter, we cannot hear you now. The microphone has been muted.
Okay, here I am again. So I would like to invite the present leader of the Global Soil Partnership, Mrs. Natalia Rodriguez, to give her opening message now. Natalia, can you hear us?
Yes, I can, but we are very lucky to count with the director of the Land and Water Division, Mr. Li-Fen Li, who will deliver the opening remarks on behalf of FAO. Yes.
Okay, thank you very much. I would have.
Thank you very much, the chair of the NETSOB, and also dear members of the NETSOB. It's a great pleasure to join you all for the 3rd annual meeting of the International Network of Soil Biodiversity. Just over 1 year ago, all the parties to the CBD, 194 countries, acknowledged that the diversity of the life is essential for all of us and all the living planet. And all the parties, they signed a new agreement, the new Global Biodiversity Framework Agreement at COP 2015, which serves as the new global strategy everyone to work on. At this historic event, soil biodiversity was really highlighted for the first time, being at a global resource that need to be preserved. With up to 60% of the species on the Earth making their home in soils either throughout their lifespan or partly. Thus, healthy soils and the ecosystem services they provide are the foundation for all the 4 betters that's consisted or highlighted in FAO's strategic framework. That's the better production, better nutrition, better environment, better life, leaving no one behind. As a global recognized mechanism to position soils in global agenda and also to promote sustainable soil management, the GSP, the Global Soil Partnership, hosted by FAO, established the International Network on Soil Biodiversity in December of 2021, the NETSOB. NETSOB is really to promote the sustainable use and conservation of soil biodiversity and to bring everyone together to form a human, and also bring all the members together through this NETSOB to form really the global thought leaders that's contributed to fulfilling the aims of the agreement that's reflected by nearly every country of the world in the CBD process. There are many challenges, certainly. One of the great challenges facing this network now is to understand the distribution and functionality of the soil biodiversity and also provide the information to help to develop policies on restoring and protecting soil health across the borders in all the countries. To meet this challenge, certainly the world need a very innovative initiatives that have to assess and monitor soil biodiversity in a harmonized way across all the national borders. And your task here as a network, as global leader on soil biodiversity, is really to build the Global Soil Biodiversity Observatory as committed at the COP15. And this observatory hopefully will monitor and forecast the conditions of soil biodiversity and also will serve as the framework to develop policies, promote sustainable soil practice, determine the economic values of soil biodiversity and build national capacities for assessing soil biodiversity and soil health. The network of soil biodiversity and also practitioners need to rise to this challenge and come together for the Global Observatory to succeed. I really appreciate all of you to following the examples, following the commitment by all the 190 parties to the CPD, and put your individual or institutional hat aside and really to look at this issue from a global perspective. And during this annual meeting, we need to also need you to represent and share the best knowledge and ideas that can really help us to advance our works on the soil biodiversity. Certainly, our soils need your great effort to promote sustainable soil management, and so as to save the soil biodiversity in the future. So for the coming days, we have a very ambitious agenda ahead of us, and I really hope that we will succeed in advancing all the activities, all the agendas, and we as well as to renew the governance of this important network. With that, I thank you very much for your contributions in advance. I really wish you a very fruitful meeting in the coming days. Thank you.
Yep, Peter, we need the room to be unmuted. Diana, yeah, Diana is taking care of that. Great. Thank you, Diana.
Thank you very much, Mr. Lee, for your opening address. I apologize for being confused, but we are very happy that you could find the time to be here and to open this annual meeting. Thanks very much again. Then we go to the next item on the agenda, and that will be the keynote lecture of this meeting. We have one, and we have found Carlos Guerra to be willing to give this keynote lecture, also on behalf of Braj Singh. And I would like to give the floor to Carlos. I already see him. Carlos, can you hear us?
Yes, I can.
Okay, then thank you very much for willing to give the keynote, and I give you the floor. Carlos, go ahead.
Thank you, Peter. I have to say that I was expecting that Braj would also give his own part of the lecture. But I can at least begin, and then if Braj can step in, that would be great. So actually, one of the things that fits both what I'm going to present here today and what Braj is also coordinating is that both of these initiatives, so both Soil1 and the crop microbiome, are global initiatives that aim to provide a basis for international cooperation. And this international cooperation comes from the multiple people that work with us, but also with the multiple activities that are attached to both these two projects. And this is what I wanted to show you here today, is what can be done when multiple people engage in one single activity, or at least one same umbrella activity that can deliver on this. So the first problem that I have is that we address this from a global perspective, but actually we try to solve local issues, and we have to have local actions in order to actually do anything to soils. And this anything to soils means that we have to do this through management, because soils will not restore themselves by them alone. At least they belong to a social ecological system, and within this social ecological system, ecology plays a role, the dynamics of the environment there play a role, but also the people that are there play a role. And we are talking about a section of biodiversity that is also usually missed. And when I'm addressing this is that when I look at soils, from my own perspective, I actually go past the physical and chemical aspects of soils and mostly look at the ecological aspect of soils that include also soil biodiversity. The problem with this is that we have a long history of trying to capture what is the pH of the soil, what is the structure of the soil, what is the carbon inside the soil. But even for those measurements, we still lack standardized procedures that are common across the world. If that's the case for aspects of the soil that have a long history already, it's even worse for an aspect of soils that does not have that long history, like the case of soil biodiversity. So one key aspect that we need to solve here is the need for these standardized procedures that go across borders of countries and across disciplines in some cases, and allow us to capture the diversity that is soils across the world. The problem with this standardization is that we then face other types of issues of international collaboration in terms of being able to share samples, being able to share even expertise across countries. And one of the relevant directives that exist in global cooperation is Nagoya. So the Nagoya Protocol is really important to maintain, but at the same time, it creates, in the case of soil in particular, it creates lots of impediments for this international cooperation that we need to solve, or at least we need to be able to pass beyond those limitations in order to have a proper description of what is soil biodiversity, because Nagoya is there to protect countries that withhold the biodiversity across the world, and to protect their interests, which is both are really important. But at the same time, we should be able to, in particular in the field that is quite new, like in the case of exploring what soil biodiversity is, we should be able to allow for the community of researchers, at least, to go beyond these protocols. The other aspect that I would like to raise is that soils have a real impact in the economy, and also that soil is private property. So it's not like if we are talking about air or water, where we can have a public domain statement where we have to protect water and soil, water and air, because independently of who owns the parcel of land. In the case of soils, this is not the case. We have to protect soils, yes, but we are aiming to protect something that does not belong to the state, in most cases, does not belong to you as a legislator or a researcher, but belongs to someone else. And this means that we have to convey the message to this someone else that protecting soils is important. And we also are in this system where the stakeholders that interact with soils are also changing. We went from having mostly family farms to have bigger companies entering the spectrum of what is produced currently, to today where we have hedge funds buying entire pieces of land, or in most cases renting large pieces of land and then hiring a company that explores the soil. This transition also means that the interests that are related to soils are also changing, because an hedge fund does not need to guarantee future development of that soil. It's renting that soil for 10 years, 20 years, and then it's leaving and renting another space, while a family farm would need to stay there in that same place. And it's easier for us to transmit the need to preserve that soil for future generations. So these changes in what is the requirements of soil and the importance of protecting soil, this is something that we need to think about quite thoroughly in terms of being able to properly protect soils. Yeah. And we should never forget that soils are also part of our culture. We have a relationship with soils, either by farming, but also by other cultural appropriation of soils, where we, for example, bury our loved ones, where we have rituals related to specific types of soils. Soils are used in painting all over the world. And all of these different aspects of soils have to be also considered when we are trying to protect them. So what is SoilBond, and what is the Soil Biodiversity Observation Network? We are a cluster of community-driven, and I would say community-driven because it's the key aspect of this, activities under the same umbrella. So we together with the GSPI, we belong to the GSPI as a community. And then within that, we are, let's say, an operational branch of this community that targets specifically monitoring of global soil biodiversity. We don't support only global projects, but we have this global perspective in place. We also have several capacity building initiatives. We work a lot on standardization of procedures, standardization of sampling activities. We do consultation with countries when a country wants to implement their own monitoring system. We also support them on that. We have activities on citizen science, for example, related to earthworms, as an example. And we work a lot on science communication, because one of the things that we easily can all get is that we can only protect what we can see. And when people traditionally look at soils, they don't see the biodiversity that is there. They see the texture, they see other aspects of soils. And we have to somehow unearth that biodiversity, those other values of soils, and show them to a broader community in order to be able to protect them. And this is also something that we try to do. At the core of our activities are what are called essential biodiversity variables. We basically try to describe them for soils. And these essential biodiversity variables are not more than a minimum set of variables that should be monitored to be able to monitor soils. In this case, soil biodiversity and soil ecology. This does not mean that this is the only thing that you should monitor. And it also doesn't mean that you should not monitor other things. It just means that if we want to have an holistic view of what the ecology of soils is, we should be able at least to monitor these aspects of soils. And these aspects of soils include several ecosystem functions related to, for example, soil respiration, to the carbon cycle, to several nutrient cycles. It also includes the monitoring of diversity per se, so number of species, the dissimilarity between communities, aspects like that. And we should also be able to monitor the habitat that allows these communities to live in. And this includes also aspects of chemistry, aspects of texture, aspects of physics of soils, but it's in the sense of trying to characterize the habitat where these communities live. You have to think about, if you're doing this for a bird, you would also try to characterize the trees that are in the forest. You would also try to characterize the climate that is in this forest, the types of features that construct this habitat that allows the bird to live. But you will also try to understand the diversity of birds, the function of these birds in this community in order to better protect it. Otherwise, we will not be able to design protection measures that align with that. This also means that we have to take advantage of proper monitoring systems. This means that from one single sample, we have to be able to extract as much information as possible. And this is something that was not traditionally looked at like this. We would have someone that would take a sample and would try to look at the chemistry of this sample. Another person would look at the physical aspects of this place, but not from one single sample, taking everything out. And this is what we are proposing, is that to maximize the effort to properly collect these samples and to properly store them in order to allow for more analysis. And I would just leave you here with what I think it's in my personal view, a good vision for the future, is to have not one global monitoring of everything, but to have scale integrated monitoring systems. This means that the countries and the researchers that also feed into these monitoring systems, these local monitoring systems, belong to a global community that allows for us to have comparable results, to have these transfer functions to be calculated. For this, we need to have samples taken locally, but also analyzed locally, but also analyzed globally. This doesn't mean all the samples, but it means that some of these samples would have to have this double sampling, if you want. We should have an independent research-driven activities. And this means that to have also a good research community that allows for these independent activities to take place. This only ensures that the quality of the data is higher, but also that the trust on this data is also higher. We have to invest in capacity building activities. We have to invest in having operational resources. There are many countries that cannot do some of these analyses because they don't have the means for that. And we have to have some kind of facilitation across countries that allows bilateral, at least, or multilateral collaborations between at least researchers within the field. And finally, I would like to say that this only works if we really promote local experts. There are many hundreds, thousands of local experts around the world that deal with the same issues every single day in their research. And we have to promote them not only locally, but also in the regional and global stage to allow them to have the resources to be able to do this in a proper way. And that's it for me. Thank you very much.
Thank you very much, Carlos. I think it's open for discussion now.
There was one question in the...
There's one question. Jacob, can you?
Hang on, I don't have the access to... There it is.
I can see the question. If you want, I can answer it.
Okay. Yes, please do so.
So the question is if so the person Talal states that renting a soil is kind of mining agriculture. Yeah, so we go to a place, we extract whatever we want, and then we leave the place for land abandonment. And he's asking, or her, she, I don't know, is asking, are there binding rules or norms to control land use and management during rental periods? And please give any examples. So to my knowledge, and so first of all, it depends a lot on the country. Yeah, so there are countries with more rules, countries with less rules, depends. But what happens is that, to my knowledge, there are no specific rules that regulate this specific type of activity yet. Because first, it's a new activity, and second, is done in a way that regulating this particular aspect, it's actually quite difficult because it's basically a company renting a place and hiring another company to come and use it. And unless we categorize this type of activity in a specific way, it's quite hard. The example that I have to give you of this happening and landing exactly in land abandonment is in the south of Portugal, where, so I'm from Portugal, and in the south of Portugal, there are massive hedge funds renting thousands of hectares to produce olive oil. And because it's a different type of culture, of agriculture also, they are basically extracting all the nutrients from the soils for periods of 10 years, and after 10 years, they just leave. They leave to the next one. And they are doing this both in the south of Portugal and the south of Spain. And there's no way currently to stop this activity because the landowners want to rent the property. They don't see the disadvantage of renting the property. They don't see the limitations that come after. So they basically just rent the property to just get their earnings for 10 years. This means the landowner, yeah? And then after 10 years, they have a piece of land to rent again. The problem is that when they are ready to rent again, 10 years later, they don't have much to rent. Yeah.
Okay, thank you, Carlos. Are there any other questions? Yes, Luca, please.
Yes, hi, Carlos. I don't know if you hear me. I hope yes.
Yes, I hear you.
Yeah, just thanks for the presentation. Just as a curiosity, I mean, you presented a nice list of very sophisticated indicators for soil biodiversity, some of them requiring quite some sophisticated technology like DNA sequencing or other top range indicators. I mean, how do you imagine to implement this in global scale, given also that this requires a huge amount of technological investment and also financial investment? Would it not be better to focus on few but easily detectable soil biodiversity features that can be detected by all of us, not necessarily needing a sophisticated laboratory for that?
Yeah. So the answer is a bit more complex. So first of all, I agree with you. So I think that we should have a set of, let's call it, minimum indicators that we should be able to calculate everywhere, to the point, just using an example of using the color of the soil, if calibrated locally, can also be an indicator for many different aspects of soil. So I would say, yes, we should have this set of minimum indicators. What I would also argue is that even in order to support those minimum set of indicators, we would need to have at least an activity that, if you want to consider this across the world, we should have at least one activity that goes across the world and collects more information than this. And this initiative doesn't have to be everywhere, but it should also allow for regional implementation. And when I say regional, I'm talking about Europe, Africa, South America. So to have regional implementations of these activities that can cluster resources and that can do this at a proper scale. If we are able to do this, then we have to face a different challenge. So in Europe, maybe we already have a tradition of collaborating and we have labs everywhere, we can do this easily. The problem is actually, in some cases, justifying resources and things like this. In a region like Africa, it's not about that. In a region like Africa, it's about finding international cooperation where a country is responsible for some aspects of this monitoring and another country is considering other aspects. For example, one with functions, one with biodiversity. That said, this doesn't solve all of this. And what I'm going to say next also doesn't solve all of this. But one thing that we would need more than this is to have proper ways of storing and archiving all these soils. Because one of the things, and Luca, you know this for sure with the JRC, is that one of the problems that we typically have is that with technology advancing fast and with us not caring or not predicting all the aspects related to archiving the soils collected 10 years, 20 years ago cannot be used today for quick assessment of timelines, for example, of trends, of things like this. So I would say that if we couple these three things, so archiving, better methods of archiving and collecting soils, funding regional activities related to international cooperation, if we have these two aspects together with a good set of minimum indicators, this would, for me, work. We don't have to have thousands of samples everywhere, monitoring everything. It would be awesome to have, but we don't have to. I agree with you.
Carlos.
Natalia, I understand there's another question. Yeah, please.
So how effectively science communication can be translated into local languages to communicate the science in a way that it can be understood without losing the content?
Yeah, so we have a good example for that. So a couple of years ago, some people from my group and now a student of mine, they created a special issue in a journal called Frontiers for Young Minds, where the objective of this issue was to pick already published papers. So the authors of those papers would then have to work on a version for kids with a kid as a reviewer with a different review style. And actually the design of the paper, the figures of the paper were all redesigned to fit the purpose of that age bracket. And age bracket here means between 10 and 15 years, yeah, of age. And what they did was they did all of that in English. So they have now 36 papers in English, translating them from scientific wording to, let's call it popular writing, yeah? And what they are now doing, what my students are now doing, is to try to translate all of those papers in as many languages as possible. And right now they transform those 36 papers in, I think now it's 200 or something, where, for example, for German, they have almost all the papers translated. We have also for Portuguese and Spanish, they have also several papers translated, but they have this in multiple languages. I don't have them from the top of my mind, but they have it. So it requires work. But again, these efforts have to be properly funded, but they cannot, I don't think that they are able to be driven top down. They have to be driven by the people that produce the knowledge and are able to spend some time in translating that knowledge to other publics, and then to have a good team of people with experience doing that. Yeah.
Okay, thank you. There's another question.
Yes.
Oh, Jacob, yeah.
Yeah, how does NETSOB reach research institutes or universities in countries across the world so far to collaborate?
Well, maybe that's a question also for you, Jacob, but on my side, I can tell you that we did this in two ways. So initially, we did this as a community activity, together with many people that are here around the meeting today. We launched a call for monitoring of soils. And the call for monitoring of soils, the idea behind it was that if you participate, everything is yours, the soil is yours, the data is yours, everything is yours. You allow this global activity to use soil data, everything. But you also gain the fact that you can also do your own publications and you can participate in our publications. This is a way to reward the scientists that collaborate in these global initiatives in a way that is meaningful for them and for their careers. This is one, let's say that for the first layer. After this, what we tried to do was to support local researchers with their own initiatives. This means that I am a Portuguese, I work in Germany, I will not compete in Uruguay for Uruguayan research funds, so I can just give them whatever I did, whatever I wrote in my own country to gain my own project, for them to get their own project. Yeah, so that's what we did. We supported them with text because I speak also Spanish. We can also, in some cases, we collaborated in their own writing of their own projects to be able to allow them to get local research funding for their own activities. Yeah, and then thirdly, what happened was that we started, because of this interaction with local universities, we also started to speak with some country at the country level with some government agencies. And what we try there is to always involve local universities. So if I speak with the Ministry of Spain, for example, this is an example, we spoke with the Ministry of Castilla y León, and we immediately engaged with our local experts in Spain for them to follow up, not us. Yeah, it's not me, I don't have to do this. But the local experts are the ones who have to be valued. Yeah.
Thank you. Any other questions? Natalia, something in the chat?
Yeah, there are a couple more.
Okay.
So there's a wonderful presentation. Africa's biggest problem is the facilities to conduct the tests. How can the international community help leverage on this challenge, either taking advantage of the already established excellent labs out there, or with equipping labs with bare minimum equipment? The former would require easing the mobility of samples between Africa and the rest of the world.
Yeah, so we do this. We actually already doing this. We also are doing this with FAO. We do this in three ways. First way is that we try to support local researchers with analysis that we can do ourselves. So let's say that we have someone engaged in soil-borne, for example, developing their own project, but they are not able to do any functional analysis, for example. We tend to offer, when we can, offer to do them in our own lab and give them the results. The second thing is that being a part of soil-borne, for example, it's not an easy thing. It's not an easy thing for the local researchers. And I'm saying this not because it's hard to sample, but it's because we have to deal with all the legal aspects of transferring samples from anywhere to, in this case, Germany or elsewhere. Yeah. So when we do this, we work in close collaboration with the local researchers to allow them to engage in this process, also facilitating, for example, the engagement between countries of the same region. Yeah. And then thirdly, this is what we are having a pilot with FAO, is that we work specifically on having capacity building locally, where we try to locally see the facilities that are there, bringing people also to other facilities across the world that we can show people different aspects of how to do things. But we try to work locally with the local labs to try to equip them as best as possible. And here is where what I was answering to Luca makes sense to me, because it's not having one single lab in Africa, for example, that does everything. Again, this would be ideal, but I don't think that's a easy path. What would be an easier path is to take advantage of the local expertise in different countries or in different regions within the same country and equip those labs to the best of their capacity for them to do their own analytics in the best way possible, but then find ways to collaborate across these different labs where samples can be managed and transported in proper ways.
Okay, next question.
So what about involving students from partner institutes in helping evolve and develop some part of NETSOB working group?
Yes. Yes, we do that, and we'll try to do that better in the future. Yes. I never ask people what are their if they're a professor or a PhD. I ask them what they want to do. So we will try to remain in that way.
Thank you. If there are not any other questions,
No?
then I propose that we follow up on the agenda. And if I look at the agenda, Raj Singh will have maybe a compliment to the lecture. Is that correct?
Yeah, I
Yes,
can.
Raj, yes, we can hear you. The floor is yours.
So following the really nice presentations from Carlos about how important the soil research and soil biodiversity is, I'm going to present the progress with these global initiatives of the sustainable agriculture and environment. You probably see there is a slight change in the name from the crop microbiome, Global Initiative of Crop Microbiome and Sustainable Agriculture to Global Initiatives of Sustainable Agriculture and Environment, just because the activity of the initiatives has slightly expanded and the names just reflect that. I will encourage all of you, when you have a time, to visit the websites. So what our key goals are, what our key deliverables are. I will briefly discuss those parts and then probably go into the activity part and give you the update where we are with the initiatives. The initiative's mission is to advance innovative farming by harnessing system approaches that accelerate deployment of nature-based solutions to sustainably increase the farm productivity, food quality, livelihood, and environment. And I just said that I'm not going to go and read everything, all aspects of that. Just mention some of the key deliverables. The key objective is quite aligned with a lot of things Carlos talked about soil-borne. That's why we work in the partnership. Some of the things NETSOB is doing in terms of the communication, and some of the things Global Soil Biodiversity Initiative have been doing for a number of years to deliver on the research and the communication side of things. So some of the key objectives are the soil biodiversity, the role of soil biodiversity in sustainable agricultures, and how that is impacted by the agronomic practices. Promote ecosystem health and climate mitigations in sustainable agriculture. Promote innovations in the soil biodiversity that promotes ultimately the soil health and the agriculture, sustainable agricultures, obviously providing the knowledge rather than driving innovation itself. And that have the advances, the socioeconomic sustainability that Carlos was just talking about, how the soil is important and soil biodiversity is important. We obviously do not work in isolations. It is simply not possible, any global initiative. So we work very closely with different part of FAO, including Global Soil Partnership, NETSOBs, Global Soil Biodiversity Initiative, soil-borne, and a number of other organizations, all chip in to deliver on the specific aspects of the initiatives. So what I'm going to talk about here very briefly, that what we are trying, and I'm guessing I can't see my headline of my own slides for some reason, but I'm hoping that you guys can see that this slide is mainly about the short-term deliverables of the initiatives, or the database of the beneficial and the harmful microbes of the soils and the plants. The database of the soil biodiversity and soil health, and how that is impacted by the agronomic practices. The linkage between the soil biodiversity and ecosystem functions, and again, how that is impacted by the agronomic practices across the globe. We want to produce, and we will produce, the global map of soil biodiversity, healthy soil, soil carbon, human pathogens, and the antimicrobial resistance genes distribution across the globes, and the drivers of those above, and the predictions based on the climate models, what it looks like in 2015 and 2080 and 2100. Initially, when we set the target, we have a really very interesting participations from the PEP pool. There was 152 groups from 78 countries signed up to it. The sample, most of the samples have already arrived in our lab. We closing that all the samples we closing all the sampling coming into our lab by February. I think we're going to settle down between 52 to 53 countries. Few disappointment, but Carlos was just talking about Africa. Initially, when we advertised, we had 21 countries signed up to. Ultimately, we got samples from 5. We probably will be lucky to get another 2. But if you look at the global survey we've done previously, this is not a bad set of data, and we probably have some things to build on. The target crops for these initiatives has been wheat, rice, corn, potato, just because they are most widely used crops across the globe. We added another crop, cottons, because cotton is used in many developing countries as a cash crop for the smallholding farmers. And so we included that as a 5th crop. The target organisms that we will measure in these samples are bacteria, fungi, viruses, fauna, mycorrhiza, human and plant pathogens, and I just talk about the antimicrobial resistant genes and the functional genes across the globes. We had a very lofty plans of to have at least 1,500 sites across the globe, and we probably wouldn't meet that. I think we probably settled down something like 1,150 to almost 200 sites across the globes. The sampling sites includes not only the soil biodiversity, but also the plants and the history of the land uses, the management practice that has been in practices for say 5 to 10 years, depending upon the farmers that provide the information, the productivity in terms of the yield, but we will also use the satellite and remote sensing data in terms of the NDVI to look at the plant productivity side of things. We want to use that information first to look at the global distributions of different microbial communities. This is just one example that we have done before, had the plant pathogens, but we will do all the soil microbial diversity, how they are currently distributed, how they are affected by the current management practices, which management practice promotes biodiversity, which management practice reduces biodiversities, and how does this looks like as the climate changes in 2050, 2080, and 2100. We will build on that information to look at those, as I said, the area of the global hotspots of the soil biodiversity, particularly in agriculture fields, and which agronomic practices has or other factors has, for example, desertifications, has led into the real damage in the soil degradations in the different parts. So that will inform the policy and land managers in terms of what area of the global hotspots for the biodiversity need to conserve, what area needs to be restored, and what are the best management practices that can help restoring those biodiversity in those regions. We will provide knowledge. We will not provide, I'm not claiming that we will provide a solution. We will provide knowledge on that. The microbial product will be developed. So example, the beneficial microbes. What are the beneficial microbes that promotes disease resistance, that promotes plant health? And those knowledge can then be used by other public and private industry for developing of the microbial products. Same goes the metabolite products, both from the plant as well as the microbial. We are doing some selected sample on the whole metabolomics to look at the metabolites that can be linked to the plant productivity and agricultural productivity. And we also think those informations will help to develop AI-based products, for example, the monitoring or the forecasting products, and improve the plant breeding program by providing those informations for the better plant productions. Obviously, those were the research outcomes. The sample analysis has already started. We believe that we should have all data in by September and October, and that will take another few months after that for the analysis. So 2025, mid-2025, probably will expect the information will be ready to be shared. But that's the research activities. There's other aspects that the initiatives does, again, in the partnership with all the organizations I was mentioning before in terms of the communications. We have launched an open access scientific journals called Journal for Sustainable Agriculture in the Environment, which have a main goal of equity and inclusivity and focus on the developing countries papers. We support engagements of multiple UN agencies. We're talking today with the NETSOB and the GSP, but FAO, but I also work with the UNCCD and CBD and other organizations. And we have just launched mid last year, the capacity building programs for how to measure biodiversity at this moment through the virtual training program. But we plan to organize some of those activities face-to-face. So for example, we are trying to organize a face-to-face training program during the ISME meeting, which will happen in South Africa in August. So we targets, if ISME agrees, to 50 people from the African continent targeted in the simple way of, say, the sequencing data to be analyzed in terms of the biodiversity, for examples. We deliver those courses through the YouTube channel. So if you want to be participating as a participant or as an instructor, please do come and join us. I guess look at other YouTube and join us both as a participant, but also as instructors who have time and expertise to give. And I will leave these presentations with everybody with clear invitations to come and join. So we all can contribute. No one persons, one initiatives, one organizations can deliver those sort of aspects. So come and join either as a research partner, as a communication partner, as a training partner. And you just have to visit our webpage, click and say join, and you have to put your name and email address, and you will be invited to contribute the way you want to contribute to the initiatives. So I'll finish with that slide. So thank you very much for your patience. If there is any questions, I'm very happy to answer. Thank you very much.
Well, thank you very much, Brash, for your presentation. Yeah, originally we have planned to break starting now, but I propose that we first take some time for questions. The break will move a little bit forward. That will not be a problem because the first items after the break will be shorter than originally scheduled. So we will have enough time for the survey. So my question is, are there any questions raised, Natalia?
Yeah,
okay.
yeah. Yes, there's one.
Jacob?
Soil fauna is a large underground zoo involving many different taxa. Which one or ones have you focused on in this initiative?
All right, so what we are doing is everything is based on the DNA analysis. So it will be mainly the microfauna and some of the mesofauna. So I guess that's the distinctions between what the Carlos doing in the soil-borne and what we are doing in these initiatives. So our analysis are based on the DNA. So it will be, as I said, the microfauna and some mesofauna.
And then would you mind just sharing the website link in the chat so that those that want to join can?
Yeah, I will definitely do that. Yeah, definitely.
Did you address all the questions? Is there not one? How I can join the partnership?
Yeah, how you can join the partnership. I think that's just on the website. Is that correct?
Yes, correct. I will put that in the chat box in a minute.
Okay, last question, Luca.
Yeah, thank you, Brajesh, for the interesting presentation. I was just puzzled by that map that you showed of soil nature hotspots, which I know comes from a publication. And I was just puzzled by seeing that the western part of Scandinavia is classified as a soil nature hotspot. So can you maybe explain a little bit better how this map was produced? Because it's quite an interesting finding.
That was a paper that Carlos is the first author. He probably is the better positions to answer that question.
Maybe Carlos can then explain, because it's really fascinating that Norway is a nature hotspot, soil nature hotspot.
If I can, I'll give the mic to Carlos in a minute. But I guess one critical component comes out of that paper, that analysis, was that there is significant mismatch. If I remember correctly, there is 39 to 40% mismatch between above ground and below ground diversity. So the concept that if you protect the above ground diversity or conserve the above ground diversity, you conserve the below ground diversity is not true. So the hotspots do vary between above and below ground diversities. With that particular reason, why that looks like maybe Carlos have a better answer
Read the paper, sorry. Maybe you should.
Don't worry, Luca. So the reasoning behind it is that we are considering nature conservation in that paper from its current status quo. That means that we do not consider biodiversity alone, and within biodiversity, you consider the presence of species plus the dissimilarity between communities. And we also consider ecosystem functions. So we consider these three aspects, so richness, if you want, dissimilarity, and function. And the hotspots that you saw in that map belong to different aspects of these three. Yeah. So you have some that are there or are present there because of community similarity. So they hold very unique communities. This is the case, for example, for South America. In many of the South American hotspots, the characteristics that are highlighted there are related to the uniqueness of these communities. We have hotspots there that are related to richness, so the amount of species there is there or it's present in a specific soil. And we have your Scandinavian example where the functions are the ones that are highlighted there. That said, there's one caveat statement here because many, I was going to say most, but no, it's not most, but many of the functions that we looked at are related to the carbon cycle. And because in Norway and the Scandinavian countries, the same thing goes for Canada, there's a lot of carbon sequestration happening. It's because of the bias of the data, it's normal that it's highlighted in that way, but it comes from this functional aspect in
Okay.
article.
Okay, thank you very much, Carlos. I noticed that there are some other questions, but again, your questions will be stored in the chat, so we don't have time to address all of them, but they can be responded later. So for now, I want to close this discussion. We start now the break of 10 minutes, 5 minutes later. So my proposal now is to reconvene at 5 minutes past 1. So we have a 10 minutes break, and then we will do the remaining items on the agenda. And again, because the first items will take not that long, we will have enough time to have a proper discussion about the survey, George, so don't worry about it. So for this, we just close the meeting for 10 minutes, and I will see you 5 minutes after 12. Thank you. Start? Okay, we will start again. Thank you very much for being here again. For the remaining of the agenda of today, we have 3 items, and we thought it's a sort of logical sequence that first, I will say shortly about NETSOB. Most of the things probably you are already aware of, but it's good to realize where we are now. And then we have a presentation about the GLOSOB initiative by Jacob Parnell, and the remaining of this meeting will be spent on discussing the survey, and the results of the survey will be presented by George Brown. So first, something shortly about our network, NETSOB. Do I have to press the button?
Yes.
So the NETSOB initiative was shortly after the Global Symposium on Soil Biodiversity by FAO, and that was held in Corona time, 2020, and about 4,000, maybe more than 4,000 people participated. So that was a fantastic, also unexpected success. So that meant that we are...
You should share your screen.
Oh.
It's sharing.
We try to... We are trying to share my screen so that everybody knows what's going on now.
I'll share the screen. No, don't share the presentation. Share the screen.
Okay, let me see.
No, one back. So again, we started after that global symposium, and that was really a success. And also because we realized that there are so many people working worldwide on soil biodiversity, we were not aware of that. So that was really something that we thought we should stay together and get a partnership, stay connected. In fact, that's the most important thing of this network, stay connected. And whether you're a scientist or a practitioner or a farmer or a policymaker, it's very valuable that we are creating a network and try to be inclusive as we can. And now we have 995 members of the network, and maybe we are over 1,000 at present. Maybe that's an old number. So we really are combining, connecting a lot of people all over the world with also a regional spread, which is very satisfactory. Now, when we started, of course, there should be some objectives, and these objectives are formulated in a quite general way, but that can also give us some room to see what we are going to do in the future and what kind of products we want to produce. So there were, in fact, 4 objectives. The first is to strengthen the knowledge about soil biodiversity. The second is to provide critical mass for the implementation of the Global Soil Biodiversity Observatory. Jacob will tell something about that in a moment. We want to contribute to monitoring of soil biodiversity status and loss. And of course, we will discuss that too today when we talk about the survey. And we will try to provide reliable evidence to support decision-making at the policy level. And also that is something that we will discuss in the meeting of tomorrow. The next slide is this. So to make things more explicit, we started 4 working groups along 4 target key products we want to produce by NETSOB. And the first is around the question how to measure it and to develop guidelines for measuring, assessing and monitoring soil biodiversity. And the second is how to manage it, how to manage soil biodiversity, and to compile a field manual about soil biodiversity management practices. And that's also an activity that we try to get a lot of people on board and see what is going on in the world, also in terms of case studies that we can learn from one another. The third is how to economically value soil biodiversity. And that's something new, and it's related to soil ecosystem services by soil biodiversity. We can learn a lot about how this is done in terms of above ground biodiversity or aquatic biodiversity. We want to make a step towards the soil biodiversity issue. And the fourth is that we want to develop effective policies in order to make a difference if it goes to governance of soil biodiversity. Now you see these four colors, and that relates to the pie diagram on the left-hand side. And there you can see that there are a lot of participants in the NETSOB network related to the first two activities, the blue and the brown one, that is about measuring and managing. And there are three more smaller parts of the pie, and that is about the third and the fourth. And there's also a lot of people, or quite some people, who are part of the partnership but have not assigned explicitly to one of these working groups. Now, to say something more about what we did so far, and I can say we are now close to finalization. We have a board meeting here now, and we're talking about documents, and we hope they can be brought out in the near future. And the first is that we conducted standardized operational procedures to assess biological soil health parameters. Of course, that is done in close cooperation with GLOSOB, and it's also based on a lot of information we have attracted by means of a global survey. And I can say, George will say more about it, but I'd just like to say there were more than 2,000 responses to that survey, which I think was also a fantastic success. You know, if you so many people who are taking the time to fill in a form and to tell us what they're doing, why they're doing, how they are doing. So that was a great piece of work. Then we are now compiling a book, a volume about collecting examples of best management practices. We focus primarily on agricultural ecosystems, and that is also a document which is now in progress. And regarding the economics and the policy, we have now, as a first step, combined the efforts of the two working groups into one, let's say, perspective review kind of document, addressing how to value economically soil biodiversity, the theoretical scientific basis of doing so, and how we can translate it into terms of effective policy measures. So these are the three documents that we are heading for, and we hope they will be finalized in the near future. Then finally, something about the governance structure of NETSOB. So in the middle, we have the NETSOB chair, well, that's me, and then we have the NETSOB coordinator. We have representatives of the Global Soil Biodiversity Initiative. We have the chair of the Intergovernmental Technical Panel on Soils of FAO-GSP, and a representative of the Convention of Biological Diversity. And under the soil, you can see four working groups, and that are the four working groups I just told you about. And there are, for each working group, we call it a vice chair and alternative vice chair. And the people who are now involved in the board the coordination is now done by Jacob Parnell and Natalia Rodriguez. The first working group is led by George Brown and Carlos Guerra. The second working group by Zoe Lindo and Jeff Battigelli. The third, economically evaluation, is by Gianluca and Giulio. And the fourth, about policy measures, by Rosalina and Luca. And I'm the chair, and I will be chairing the work until tomorrow, 12 o'clock, because then we will elect a new chair. So again, if you want to be part of that process, stay connected or be connected at 12 o'clock when we will doing this. And this is for it. This is shortly from what is NETSOB about. And that is a sort of startup presentation to give Jacob the floor about telling us something about the GLOSOB initiative. Jacob, You need this computer?
Yes.
I will wait here. You sit in the chair. Okay.
Let me try this again.
Oh, there it is. Okay. So I'm gonna talk a little bit about the Global Soil Biodiversity Observatory. So at the last, the Convention of the Parties, the Convention on Biological Diversity, as mentioned earlier by Director Lee, 193 countries came together and said that we need to be measuring biodiversity. And so they've tasked NETSOB and the FAO with trying to figure out how we're going to do that in a global context. And so the goal is to be able to monitor, forecast the condition of global soil biodiversity. And so we've got kind of how we're going to do that and what that entails. And really, a lot of this is kind of along what Carlos and Brajesh were talking about earlier, is we want to be able to have indicators that can be measured anywhere, so that these are ubiquitous indicators that can be measured, and we want them to be measurements that individual countries can adopt. So whether they choose to join into kind of a regional area or not, we want them to be able to say, these are the, we want to join this GLOSOB membership, and we are going to build the capacity in that country to be able to measure those measurements. And so there will be a lot of opportunity to have local and national experts to be able to contribute to the Global Soil Biodiversity Observatory. We obviously want to have indigenous people involved in this process of developing this Global Soil Biodiversity Observatory in these local areas. And so I kind of wanted to show you this roadmap. So this is the first thing we need to do is define these essential biodiversity measurements. And we want to have 3 different tiers. So there's been a lot of discussion about, what happens if you're a country that doesn't have the capacity to be able to, collect all of these measurements. And the idea is that there is a limited membership, this lower tier where, GLOSOB through GLOSOLAN, through FAO, through some of these initiatives will be able to help build the capacity to measure these bioindicators at this limited level. And then that country can start collecting those measurements almost immediately, while we continue to work with them to build the capacity for this moderate membership and this full membership, so that we're immediately collecting the information, and then we're building capacity to collect more information. And so I will kind of share this little bit here in a minute of the essential biodiversity measurements that we're proposing, as well as these different tiers. And then the idea is that we take this to, and we assess the national capacities. So what is the infrastructure? One of the questions earlier mentioned some countries that might not have the infrastructure to do this. So we need to go find out the infrastructure, provide the materials and the infrastructure to be able to collect the measurements and build that capacity. And we also need to be able to find those national hotspots. So working with local experts, with those governments, with indigenous groups to be able to find out, where are the areas where we should be collecting the biodiversity measurements. And then kind of the next phase of this would be to actually start collecting those measurements. And so this is where we build the capacity, we acquire the equipment, build the training, secure funding from those governments to be able to, either from those governments or from other governments to help sponsor to get this project moving, and we actually begin monitoring. And then, as I said earlier, while all of this is going, we then continue to build the capacity to increase that membership, to increase the bandwidth of those countries to be able to strengthen their monitoring systems or establish it if needed, and then to be able to advance that and to be able to, the goal of this in the end is to be able to provide those countries with kind of an idea of what the status of biodiversity in their country is, and so that they can then use that to make key informed decisions on how to manage the soils and the biodiversity within their country. So I kind of went through that quickly because I really wanted to show this, and it's quite a bit. And so I'll leave it on this for a little while, but this right now is the proposed, these are the proposed measurements. So the bioindicators, these are bioindicators that are used by SoilBon. And then we've got this kind of, how are we going to measure those bioindicators? And then we've also got that membership tier. And so most of the limited membership tier, the measurements that you can see that are bolded have actually already been, those SOPs have already been developed by GLOSOB. And so those are being, the capacity for those are available now and are being built by GLOSOB. And so you can see that most of those under the limited membership tier have already got those SOPs. And so then as far as NETSOB, we would help GLOSOB contribute to those that do not have those harmonized SOPs yet, be able to build those, and then we would work with GLOSOB to be able to train those locally, build those capacities to be able to get those measurements. And then back to, Luca has mentioned a couple of times about, these expensive DNA type of, and that I agree with him completely. And I think the answer is, instead of trying to work to the lowest, it's to bring everybody up to that level. So it's start with, so we start with what they can do, and then we build the capacity for them to be able to do everything that kind of these more advanced technologies and techniques for measuring soil biodiversity, and build the capacity locally for those governments. So I wanted everybody to be able to look at this and then kind of leave it for, I think this is actually the last slide I have, so that everybody can look at it, and because ultimately what I want to come out of this is for us to be able to say, yes, this is the way forward. And then if that is so, we'll have the NETSOB board approve these measurements and this strategy moving forward. And then as FAO, we can start moving on actions towards this. So I guess I would open it up for questions.
Are there questions? Jacob, is that okay?
This is from Jacob, right?
This was all, it was from Brad.
So there are no questions for Jacob yet.
Okay.
Anyone from the room who have a question for Jacob? If there might turn up questions, they're stored in the chat, so don't worry. And then we just continue with the agenda. And that brings us to George Brown, who will tell us about the global survey, which was conducted on soil biodiversity. George, please. The floor is yours.
Okay. Here.
Is there a question? Okay, let's do that then. George, you can stay connected. Jacob, there's one question. At what depth are you going to sample? 10 or 20 centimeters?
So we're working with GLOSOLAN. I think most of GLOSOLAN is from 0 to about 15 centimeters depth. And so right now we're looking at that. Again, all of these protocols that haven't been developed yet are hopefully being developed with some of you. Some NETSOB members are involved with some of these SOPs that are being developed. We would like it so that they're not having to go and sample at different depths for different measurements. So ideally, we would want to stick to kind of a 0 to 15 centimeter depth for all of the samples. We'll have to see if there are any problems.
Also, if you think about different land uses,
yeah,
stick to one depth. Okay. Then the second question comes from our student restricted from the mentorship.
Okay, yeah, that's a good question. So as far as the membership goes, the membership is for countries, and personally, I don't see it limited to just countries. I can see, for example, we know that a lot of some of the biotech companies are trying to get involved with soil health, and so I can see, a company saying, well, we've got fields in South America, North America, and Europe. We want those tested for soil biodiversity, and we're going to use these. They can be membership. So this isn't a student or even a university. This is more a government kind of level membership. This, remember, is a response to those 193 countries that said, we think biodiversity is important. Now tell us how we're going to measure this and how we can do this. And so this is our response to that, saying this is how you can start measuring it immediately.
Good. The next question, Jacob, is about the 16S and 18S analysis. You see it?
It would be good to have 16S and 18S analysis in limited requirements. Otherwise, we have no biodiversity measures. So
it's more a remark.
So yeah, that's suggesting moving it from kind of a moderate membership to a very basic membership. And I think I mean, I'm a microbial ecologist, so I agree entirely with that. But we also have to remember that there are some places where the capacity does not exist right now, even for bringing back, preserved samples back to the lab to do DNA extraction and analysis. And so, I agree, and I mean that's something that we need to discuss. But there are currently places that do not have sequencing machinery, and the whole goal of this is to make it so that if the country decides to, they can do it entirely locally. So the labs that are there within the country can do everything.
Okay, let's do another 3, right? And then we go to George. So the first is about, please to be careful with the CO2 change. You could end up with no useful sequences. Can you say something about that?
So yeah, this is for the for measuring kind of DNA from larger remnant type of animals that have left their mark, so to speak, in the soil. And yeah, this is still quite experimental, although so right now there isn't or there are methods where you can measure like mammal and avian DNA, or you can measure animals and birds and things like that, but that's a lot of capture and things that are that go a little bit beyond what NETSOB would be able to build the capacity. And so this is kind of a proxy of being able to find out what are the animal type of DNA. And yes, okay, there would definitely have to be some caveats around that.
And it seems to be a long discussion. And then, of course, a very important question is also how the soil bomb fits into GLOSOB, if I understand that question.
So I think that soil bond and GLOSOB can be very, very complementary. Right now, I think that, and I see us as using like GLOSOB as hopefully being able to adopt some of soil bond's already developed protocols. Soil bond also has some capacity building programs, and so potentially what we could offer for some of these countries is to just say, well, why don't we bring in soil bond to help build the capacity to train, you know, your laboratory technicians on these methods, to build it up, to help you with the equipment and everything for these GLOSOB measurements, and for, you know, countries that are already, you know, involved with soil bomb projects, they might just be able to say, well, we already have some soil biodiversity hotspots. Maybe we'll add a couple others in more agricultural areas or urban areas potentially. And then, you know, we are already doing all of the measurements that are required for suggestion.
Okay, and the last question we will address now is, do community level physiological things, profiles, are those included, right? And the rest we will address later. Do community level physiological profile included in the soil respiration? Jacob, can you say something about that?
So I'm not sure I understand entirely the question. We are measuring soil respiration rate, but I think it's important that soil respiration is not measured alone. So it has to accompany other things.
Okay, well then, thank you very much, Jacob. Also, thank you all for having these questions. The remaining are stored in the chat. And because of time, we want to continue now with the next item of the agenda. George, are you ready to stay connected? Okay, then the floor is yours. George. Okay.
Well, thank you very much for all of you who are present online and in person here. It's a pleasure to be here speaking with you once again about the results of the survey. Thank you, FAO and GSP, for allowing us to be here in person. This is great to be in a small group here, and great to be sharing these results as well. So first of all, I thank then the FAO and GSP, but also the large number of co-authors and people who have helped us develop these results, and particularly all of those who answered the survey, with which we wouldn't have these results. This survey was applied in 2022 for 3 weeks. The 70,000 plus emails that were sent out were obtained mainly from the literature, but also from the Global Soil Biodiversity Symposium, which had over 5,000 emails from the FAO, of which received almost 2,700 replies. That's quite a low amount, percentage of replies, but understandable considering COVID, and all the various problems associated with a long survey, which had 122 questions, although you didn't have to answer all of them. Very few people actually did. Most people answered probably only 30 to 40 questions, so less than a third of the survey. It had 11 sections, of which there were some devoted to the taxa, so microbes, the fauna, and then some devoted to the functional levels and assessments of the community biodiversity, as well as biodiversity inventories.
The problem with the micro. See in the chat. Is that correct?
Can you hear me? Or do I need to
sit here? Maybe this one works better. I don't know.
Or I can unmute myself. Nope, I will get microphony. Can you hear me now? Is that any better? So, anyway.
Seems to be an echo. Maybe I have to turn this off.
Yeah,
Jacob?
turn that one off.
Okay.
So we had some aspects related to if countries were doing inventories or monitoring, who was studying ecosystem services or applications of soil biodiversity in practice, and the various threats to soil biodiversity, as well as finally education, communication activities, and public policies related to soil biodiversity. Now I can't move. So the main objective of this was to really get a status quo, state of the art, who's doing what, where, and how in terms of soil biodiversity, particularly inventories, monitoring, mapping, and various databases, information on particular taxa and the functions, how these were actually being studied, which methods were being used, which were the main uses of soil biodiversity, the work on ecosystem services and valuations associated with soil biodiversity, and the presence and knowledge on public policies related to soil biodiversity. So 139 countries replied, over 1,300 institutions. Most of them were from Europe, so almost 40% of these respondents were from Europe, and then the highest number was followed by Asia, and then 12 to 15, 17% were from Africa and from Americas, few from Oceania, and actually only a few countries had over 100 respondents. That's Italy, India, Brazil, and USA. There was not a great gender balance, so it was about 60-40, meaning more male than female respondents. Most of the respondents actually worked in educational institutions, also people in government institutions, and most of them in research. Many of those people were actually focusing on local and national level studies, less on global studies. The main themes were focused on microbes. So most of the respondents, almost 68-7%, worked with microbes, a lot less with fauna. Still a high proportion working with functions and actual applications or uses, ecosystem services related to the soil biota. Quite a few of them were involved in inventories. And if you look at this pie diagram on the right, most of the people worked with only one taxonomic level, and very few people worked with more than one taxonomic level. As you get more and more taxon or groups working at, there was a fewer proportion of people who had a more systemic approach to soil biodiversity. The land uses covered involved primarily anthropogenic ecosystems where crops dominated, reaching over 50% of the respondents. Natural systems were almost always used as reference points, so between 30 and 40% of them were using native grasslands or native vegetation. Degraded lands, forest plantations were less studied, and even less pastoral reclaimed sites or urban areas. In terms of inventories worldwide, 50 countries identified that they were doing some kind of inventory. That doesn't mean inventory of all taxa, it just means inventory of a particular organism or particular taxa or particular function. And so it's the map showing, again, concentration of countries in Europe, North America, and Asia, less in Africa and South America. Take-home messages from here, where we look at the inventories, we find that quite a few respondents actually didn't know if there were inventories in their countries. And when they were aware of them, most of them said they studied microbes. So almost 80% of the people of these inventories focused on microbes, a lot less on fauna, but still close to 50%. So still quite a bit in terms of macro, meso, and microfauna, a lot less with the vertebrate organisms. When you look at the monitoring programs, this looks a lot like the inventory, and these meaning monitoring, once again, of any taxa or any level of function, where, again, European countries and Asia and North America were prevalent, and less in African and Latin American countries. And when you look at the level of these monitoring programs, most of them were at the national level, less at the local or state level. And just as the inventories, microbes dominated, there have been 66% of the respondents said they studied microbes, and a lot less attention devoted to fauna. When we look at the microbial part of the survey, people who actually did work with microbes, most of the respondents said they worked with bacteria and fungi, so between 80 and 85%. And this isn't surprising concerning the importance in soils, the historically most studied organisms, their well-established taxa with molecular markers and so forth. So a lot less people dedicated time to studying virus, algae, or lichens. So these were our taxa that were less evaluated. I'm not going to spend a lot of time going through these methods because this is something that's going to be shared again tomorrow as we look at the methods and what's been done by working group 1. But I just wanted to highlight briefly some of the main methods for microbes and for fauna. Again, in terms of culture-dependent methods, molecular and phenotypic were the most used. 67 to 76% of the people were using these, which is common. All other methods were less used. And in terms of the molecular, well, most of them were using genome sequencing and fingerprinting. When you look at the culture-independent methods, most of these, of course, were based on molecular, so metagenomics, high-throughput sequencing, metabarcoding, metagenomic, qPCR, et cetera. So these were the most used methods for these. And of course, then, fingerprinting and 16S RNA, ITS for fungi, et cetera. So these more common methods. And of course, most of the people use the Illumina as the metabarcoding method. When you look at the activities and the processes, so what people were actually looking at in terms of what the microbes are doing in soils, respiration and enzymatic activity were the most evaluated. So these were over 60%. And then when you look at some of the other methods, these were mainly nitrogen mineralization, mycorrhizal colonization, and decomposition. These were less common, but still quite prevalent. And then, of course, when you look at greenhouse gas emissions and other aspects, these were less evaluated. In terms of microbial biomass, well, fumigation extraction is the main method. The others were less used, so SIR and fumigation incubation. And when you look at the applications of these microbes, so what people are studying in terms of what these microbes are purposes of serving in the soils, these are mainly related to plant growth promotion, biological nitrogen fixation, phosphorus solubilization, so practical uses. Then, of course, either uses bioindicators for inventories, and then for checking bioremediation sites, and then for biological control. When you look at the microfauna and the mesofauna and the macrofauna and megafauna, so starting with the microfauna, nematodes, they're all over the place. They're probably most abundant invertebrate in the world. They were the most looked at. So most of the focus was on free-living nematodes and plant parasitic nematodes, less on the other groups. Protozoa was less studied, and then the other two groups, tardigrades and rotifers, even less. most of the nematodes by Behrman funnel extractors, protozoa, and various methods. And what was highlighted here is that actually only about a fourth of the respondents actually were active in taxonomy, so identification of the microfauna. In the mesofauna, we looked at, most of the studies had people doing mites and springtails, which of course are the most abundant groups anyway. Then again, followed by beetles and ants, and all others less than 18% of the respondents. Only about a quarter of the people were actually studying the whole community, so devoting their time to assessing various taxa. Most of these were looked at with Berlese or Tolgren funnels, so extractors in the lab, and around a third of the people were using pitfall traps. And again, a third of these respondents actually active in taxonomy. When we look at the macrofauna, most of the study were on the sort of the more charismatic ecosystem engineers, so then termites, ants, earthworms, beetles, etc. And then again with predators and detritivores also being more studied, less focus on phytophages amazingly, and only around 18% of those respondents actually looking at the whole community. Most of these studies were done using hand sorting, so that was really the best and most commonly used method, or indirectly with pitfall traps. And about a third of the people were active in taxonomy, identifying these organisms. When you look at the megafauna, these were few respondents overall, so very few people focused on megafauna, but when they did, most of their studies were involving rodents, less with birds and snakes that have some influence in the soil. The methods were very taxon dependent, and only about 19% of those were active in taxonomy. So when you look at the applications of soil fauna studies, so what people are looking at in terms of what they do, what are their purposes in the soil, again, some of these were shared with microbes, so the bioindicators with nutrient cycling, plant growth promotion, inventories. There were some people using composting and monitoring inoculation of these organisms, and of course, biological control. When you look at the methods to evaluate the functions and what these microbes or fauna were doing in the soil, a lot of people were using molecular methods quite frequently, and then decomposition as another main function that was being evaluated. And a lot less people were using more sort of semi-field or food web or trait approaches. And very few people were actually using ecotoxicology or bioturbation as a means to evaluate the functions. When you look at the services that these people were studying and how many people were involved in evaluating this, about half of the respondents were studying ecosystem services, which is really amazing. Most of the people who were actually involved in evaluation were using market-based methods, a lot less with the other techniques, so really preference or declared preference. When you look at the services people were looking at, most of the services studied involved nutrient cycling or biodiversity conservation, climate-related variables, erosion or flood control, pathogenesis, waste recycling, or soil as a habitat. So various functions were being looked at, various ecosystem services, and the most common indicators of these ecosystem services that were being used to evaluate were microbes or microbial biomass, and diversity indices and aspects related to nutrient cycling or decomposition, mineralization. A lot less focus on functional traits or primary production, amazingly, and then less on other services like enzymes, greenhouse gases, etc. And a lot less people were using soil fauna to evaluate as indicators. When you look finally at the final part of the survey, where, well, what sort of knowledge do people have about public policies, or what's available in terms of conservation efforts? Actually, most people had very little knowledge about legal frameworks or public policies, or even international instruments or national instruments. So the take-home message here is actually there was very little knowledge among the respondents in terms of what is available in, regarding public policies to protect or conserve soil biodiversity. When they did know about it, the focus in terms of public policies seemed to be a lot more directed to sustainable use or to conservation. but less to its actual protection of soil and its biodiversity. So the conclusions that we have from this survey so far is that most of the soil biodiversity studies and people involved in this are government-based or educational research scientists. My main focus has been on microbes in terms of versus fauna. There's very few active taxonomists for all of the taxa. Overall, there seems to be a focus more on functions rather than on taxonomy. There was a maximum of 50 countries that identified having inventories or monitoring programs, and most of those focused on microbes and not on fauna. And very few of them had a wide range of taxa involved in their monitoring or inventories. The ecosystem studies are quite popular, and there's a wide range of indicators being used for evaluating these. But overall, in terms of public policies and frameworks, there was very little knowledge about what's available for these purposes. So thank you for your attention. That's it.
Thank you very much, George. We can all see that this has been a huge amount of work. And we are really grateful, you and your group, for doing this important piece of work. Thank you very much. We really appreciate it. We have still some room for questions. Jacob, can you guide us through the questions?
So here's one. I teach soil biology and supervise students in related research. We do some work on earthworms and how they're influenced by land use and land cover changes, including agronomic practices like use of pesticides and fertilizers. Our RISA lab can do rough work on bacteria, fungi, and actinomycetes. There are challenges with capacity and equipment to conduct studies on nematodes, mites, columbella, etc. To come up with a meaningful suite of indicators to infer impact of human activity on trophic levels, function, and services of soil organisms, it is important to focus on tools and techniques that enable a range of these organisms to be analyzed from one sampling. I'm not sure if there was a question in there.
I agree.
There's a question also on top of that.
Yes.
Oh yes, was the survey offered in multiple languages?
No, the survey was offered only in English.
And what is the percentage of these 50% or 50 countries from total countries?
There were 139 countries replied. So of those 139 countries, 50 said they had monitoring programs or inventory. So it's about a third, a little less than a third, 40%.
All right, you dots, you can check on this. I think that's all of the.
Okay. And there's one remark, I think. Thanks, congratulations, George, for being the champion of using international data standards. Well, take that as a compliment. I think that is a last chat.
There is one question in here.
Okay, we take that one too then.
What are the data standard systems you envision to support the NETSOB working group 1? It says GBIF is of course available to continue collaboration.
I think that's a great question. In fact, that's one of the aspects of GLOSOB that we have not yet solved or resolved. And this is something in terms of data ownership and data sharing and data use, which will require considerable meditation and obviously always recognition of the main contributors, so that they know they are being recognized for their efforts. So I think that's obviously a premise which we will keep, and as much as possible obeying the rules of fair and free, openly accessible, etc. But also always recognizing the data and the original contributors.
Do you have time for another couple questions?
I think we can do Yes, we can do another 2.
Okay. What would be the consequences of the results of your survey for future focus on NETSOB? So which gaps or needs should be addressed specifically?
Yes, great question. Yeah, we definitely identified that there's, and this, we complemented this survey actually with the literature review, which I haven't presented here. I will present some results of that literature review tomorrow, which actually shows a lot of the same similar results. There's a major gap in terms of in terms of knowledge, at least of public policies. This obviously shows there's a lot of effort with microbes and less with fauna. There's a huge suite of indicators to be used, and yet already mentioned today the difficulty of how to apply those worldwide and how we can make these results and techniques more available and more usable worldwide. And then, of course, a minimum number of methods which can be actually compared and shared and used worldwide that allow for comparison between all the countries and all the participants. So I think there's quite a few gaps which we've identified with the survey, which allow us to prioritize the actions of the network.
You have 2 minutes to say something about the last 2 questions. Maybe the first one we can also pass to tomorrow.
Yes, the little bit about public policy on soil biodiversity, we'll actually be discussing that tomorrow.
If you're on then again, then we will discuss that for sake of time. And then the final question, something close to your heart, George. You can answer that maybe.
Yes, so fauna, great to see that hand sorting is widely used and works well, as it did not work so far in our focus area of alpine compacted soils. The pitfall traps work great for us, and we'll continue trying out hand sorting.
More or less a remark, but
yes,
nice to hear.
Thank you. Yeah, that's great.
Okay, well, I would suggest that we close here by this discussion. And we are also now at the end of the first part of our annual meeting. We continue the meeting tomorrow at 12 o'clock. And again, for those who have missed that, the agenda of tomorrow is changed. So we start with the item election of the new chair. So if you want to be part of that process, please connect at 12 o'clock, because then we will do that agenda. And the remaining of the agenda is that we get reports of the 4 working groups, so you can also take advantage of that. And hereby I would like to close this session. And thank you very much. Thank you very much for joining us. And I hope we will meet again tomorrow. Thank you. Bye.